What if I told you that a single missing data point on a chart is essentially the reason a space shuttle exploded?
It's a terrifying thought, honestly.
Right. I mean, in January 1986, the Challenger disaster just completely shocked the world. And it ultimately came down to this catastrophic failure in how data was presented and interpreted.
Yeah, the engineers actually had the numbers that could have grounded the flight.
Exactly. But the way they graphed those numbers, it completely hid the very danger they were trying to expose. And well, we are going to dig into exactly how that happened today because we're pulling from a massive stack of sources on interpreting scientific data for this deep dive.
And we're specifically looking at study materials tailored for, nursing entrance exams like the TEAS and the HESI.
Yeah, and the stakes attached to those exams are incredibly high for anyone entering medical field.
Oh, absolutely. But mastering data interpretation, it goes far beyond just clearing a hurdle for nursing school. We are talking about a vital, literally life or death skill here.
For sure, because every day we are just bombarded with charts about pandemics or, climate change or even just those flashy ads for fitness supplements on social media.
Right. And when you learn how to properly read results and analyze tables and graphs from experiments, you are essentially developing a superpower.
Right.
It prevents you from being easily fooled by manipulated numbers.
A superpower. I like that. So our mission today is to break down exactly how you build that superpower. Looking through these sources, interpreting data isn't just one single action.
No, not at all.
It's actually the intersection of three distinct steps. First, there's reading, like literally locating the values, identifying the axes, checking the units.
Yeah, just the baseline facts.
Right. And then second is reasoning. That's recognizing the pattern, whether a line is shooting straight up or flattening out. And third is evaluating.
Which is really the critical thinking part.
Exactly. Asking the tough questions about whether that pattern reflects a real relationship or if it's just a complete coincidence, or maybe a fundamental flaw in how the experiment was built.
But before we jump into how to navigate those steps on an exam, we really need to look at how this skill was even invented. Because for a huge portion of human history, interpreting data simply wasn't a thing people did.
Wait, really? Not at all.
No. If you go back to the 1600s and 1700s, data tables were basically used like illustrations in a storybook.
Oh, wow.
Yeah, they were just decorative elements. They were thrown in to back up a philosophical claim the author had already established in the text. They weren't really meant to be analyzed on their own.
Okay, let's unpack this because that is wild. Think of early data like the illustrations in a storybook, just there to look pretty, versus modern data being the actual text of the story itself. The numbers were just window dressing.
It's crazy to think about, considering how totally data-obsessed we are now.
Seriously. But the sources highlight Francis Bacon in 1620 as a major turning point. He wrote a book called Novum Organum and argued for something incredibly radical at the time, which was systematic observation.
Right, he was pushing back against scholastic reasoning.
Yeah, which was basically the academic habit of just blindly trusting ancient philosophers. Like, people used to just say, well, Aristotle said heavy things fall faster, so it must be true.
And Bacon argued that, no, we actually need to drop things, observe them, and record the numbers.
Which seems so obvious to us now.
It does. But that insistence on prioritizing real-world observation over ancient authority, that is the direct ancestor of the modern data table.
Even with Bacon's push, though, those early observations were often buried in dense prose or just massive, totally unreadable grids of numbers.
Yeah, the visual communication side took a long time to catch up. It actually took until 1786.
Okay, so a Scottish engineer named William Playfair steps in, right?
Exactly. He invented both the line graph and the bar chart. He was trying to show Britain's really complex trade balances, and his brilliant insight was that a trend taking pages of text to explain could be made instantly visible in a single image.
He turned the data itself into the argument, but just putting numbers on a graph didn't solve everything, did it?
No, because people could look at the exact same line and argue for hours over what it actually proved.
Right, and that ambiguity led directly to the statistical revolution in the late 1800s. We see Carl Pearson popping up in the research here. In 1896, he formalized the correlation coefficient.
Which was huge. Instead of scientists just eyeballing a scatter plot and kind of debating whether the dots look connected, Pearson created a math formula. It assigns a strict score to the relationship.
And the mechanics of that formula are fascinating. It calculates how tightly clustered the data points are along a line, scoring the relationship from -1 to +1.
Right, so a score of exactly 1 means a perfect lockstep relationship. As one variable goes up, the other goes up proportionally.
And a score of 0 means it's just complete random static. By mathematically quantifying the relationship, Pearson basically allowed scientists to stop relying on visual guesswork.
And then Ronald Fisher steps in around 1925 to build the whole modern framework of controlled experiments. He was getting really frustrated by scientists publishing dramatic conclusions based on tiny, uncontrolled samples.
So he set the rules for randomizing subjects and using control groups.
Exactly, so that the data you collect actually means something. We also have to highlight John Tukey in 1977. He brought in the modern ethos of exploratory data analysis.
Which means what, exactly?
Well, before Tukey, the culture of science was heavily focused on testing rigid preset hypotheses. He argued that we were bringing way too much bias to the table. His philosophy was that you need to just plot the data and explore it visually before you ever assume what it should show. You have to let the numbers speak first.
That makes a lot of sense. So thanks to pioneers like Playfair, Pearson, Fisher, and Tukey, we have these incredible visual tools. But history shows that the human brain is naturally quite bad at reading them instinctively.
We really are.
Yeah. So if you are staring down a TEAS or HESI A2 nursing exam, you have to master the core mechanics, the first principles of graphs. And the absolute foundation is the axis. Every single graph has 2 questions built into it, represented by the horizontal x-axis and the vertical y-axis.
And misidentifying those axes is the absolute fastest way to fail a science question. The x-axis is always the independent variable. This is the condition that the experimenter is deliberately changing or manipulating.
Right. And the e-axis is the dependent variable. That is the measured result or the outcome of the experiment.
Yeah.
I always try to think of this using a factory machine analogy. Think of the x-axis as the physical dial you turn on the machine. You completely control the input. And the y-axis is the number that pops out on the digital screen on the other side.
Oh, that's a great way to look at it.
Thanks. Yeah, you don't control the screen, you just read what happens after you turn the dial.
Mm.
If you swap those on an exam, you are essentially claiming that the digital screen turning on caused your hand to move the dial.
Right, you have the causality completely backwards. So once you have the input and output straight, the next core mechanic is reading the shape of the data.
Because a shape isn't just a picture, right?
Exactly. The shape of a graph is inherently a claim about a relationship. So a linear relationship is a straight, rising line. It indicates a constant rate of increase. For every unit you move on the x-axis, the axis goes up by the exact same amount every single time.
But if you see an exponential shape, the rules completely change. That's a curve that starts off rising very slowly, almost flat, and then suddenly shoots straight up into the sky.
Which we saw constantly on the news with unchecked infectious disease outbreaks. The rate of the increase is itself increasing. It's not adding 10 new cases a day. It's doubling every day. 2 becomes 4, 4 becomes 16, 16 becomes 256.
Wow, yeah. And you also need to recognize a plateau. This is a line that rises steadily, but then abruptly flattens out horizontally.
In the biological sciences tested on these exams, a plateau describes a system approaching a limit, like a population of animals reaching the carrying capacity of their habitat. They reproduce quickly until they run out of food or space, and the population growth just flatlines.
You also see it in chemistry, right? When an enzyme reaction reaches saturation, you can add as much substrate as you want, but the reaction just cannot go any faster.
Exactly.
Okay, so those shapes make intuitive sense, but the sources point out something totally counterintuitive, and that's the cloud of points.
Ah, yes.
This is a scatter plot that just looks like someone sneezed dots onto the page. There is absolutely no visible pattern, no line, no curve. My instinct is to look at that and think the experiment was a complete failure because they didn't find anything.
Wait, so finding absolutely nothing is actually a major scientific victory?
What's fascinating here is that, yes, it actually is. That is a very common misconception. Finding absolutely nothing in a scatter plot is a massive victory. Recognizing no pattern is a highly valuable, testable finding.
Because it proves there's no link. Exactly. It provides concrete evidence of no relationship between your independent and dependent variables. If you are testing a new medication to see if it lowers blood pressure and your scatter plot is a random cloud of points, you just proved the drug doesn't work. Ruling out a connection is just as vital to medical science as finding one.
Okay, so we have the baseline mechanics, the axes, and the shapes. Now we really need to look at what happens when we apply these tools to actual raw numbers, because the exam materials emphasize the power of the data table.
Tables reward active computation, not just passive reading. If you approach a data table like a paragraph of text, you are going to myth the hidden variables. You have to interrogate the intervals between the numbers.
Right, so imagine a classic exam question where a plant's height is measured every 3 days. Day 0, it's 2 centimeters tall. Day 3, it's 5 centimeters. Day 6, it's 11. Day 9, it's 14.
Okay, I'm tracking.
A superficial reading just tells you the plant got taller over 9 days, but if you actually pull out your pencil and calculate the intervals, a completely different story emerges.
Let's run the math on that. From day 0 to day 3, the plant grew 3 centimeters. From day 3 to day 6, it grew 6 centimeters. And from day 6 to day 9, it dropped back down to 3 centimeters of growth.
Exactly. By doing that simple subtraction, you reveal a nuance that the raw numbers totally obscure. The plant's growth accelerated dramatically in the middle of the week, and then it slowed down.
And if the multiple choice question asks you to identify the period of peak growth, you only get the right answer if you actively compute those intervals.
Missing the nuance in a table is how you fail an exam. But in the real world, it has massive consequences. Which brings us to one of the greatest triumphs of data interpretation in medical history.
James Linn's scurvy trial in 1747.
Yes, scurvy was absolutely devastating naval crews. Lynn took just 12 sick sailors, kept them in the exact same environment on the exact same basic diet, and divided them into pairs. Each pair got a different daily supplement: vinegar, seawater, cider, and one pair got 2 oranges and a lemon.
And within a week, the citrus pair was essentially cured and helping nurse the others, while the rest were still dying.
A sample size of 12 is incredibly tiny, but Lynn's experiment changed the world. And it worked entirely because of the control. He held every single variable constant: the damp air, the salty food, the ship's rocking, everything except for the independent variable he was testing.
Because he controlled the environment, the pattern in his data table could be definitively linked to the citrus. Another massive triumph from the sources is John Snow's cholera map in 1854.
Right, cholera was tearing through London, killing thousands. And the dominant medical consensus was the miasma theory,
Which blamed the disease on invisible clouds of bad air.
which is just terrifying to think about. But Snow didn't just argue with the experts. He systematically plotted every single cholera death as a black bar on a street map of the city.
And if the disease was caused by a giant cloud of bad air, those deaths should have been spread out relatively evenly across the neighborhood. But that's not what the data showed.
No, the deaths formed a severe, tight spatial cluster right around a single location, the Broad Street water pump. Snow used that visual pattern to prove the disease was waterborne. He convinced officials to remove the handle from the pump, and the local outbreak completely collapsed.
But the tragic counterpart to Lyndon Snow is what happens when data is mishandled, which brings us back to the Challenger disaster we mentioned at the start.
Yeah.
7 astronauts lost their lives because an O-ring seal failed in unseasonably cold weather. The heartbreaking part is that the engineers suspected the cold was dangerous. They created a chart plotting O-ring damage from previous shuttle flights against the temperature at launch, trying to convince management to delay the liftoff.
But they made a fatal error in how they constructed their data set. They only plotted the flights that had experienced damage. He intentionally excluded all the successful flights from the chart because they thought those data points weren't relevant to showing a failure. Here's where it gets really interesting, though. Is this really a failure of understanding graphs, or was it a failure of human psychology and corporate pressure?
It's a mix. But the tool the engineers used to try and persuade management was fundamentally broken. By excluding the successes, it completely erased the baseline.
Wow.
Yeah. Edward Tufte, a pioneer in data visualization, later went back and plotted the full data set, all 24 previous launches, including the successful ones. When you look at the complete graph, every single successful flight without damage occurred in warm weather, and almost every flight with damage occurred in cooler weather.
The correlation is undeniable.
Exactly. But by excluding the non-events, the engineer's original chart just looked like a random scattering of dots. Management looked at a flawed graph, saw no clear pattern, and ordered the launch.
You literally cannot prove that cold causes failure if you hide the data showing that warmth is associated with success. Leaving out data is profoundly dangerous. And, well, that transitions us straight into the dark side of data: intentional manipulation and the classic traps that trip up everyone from nursing students to tenured scientists.
The most notorious modern example of this is Andrew Wakefield. In 1998, he published a paper in The Lancet claiming to find a link between the MMR vaccine and autism. This paper sparked a global panic, but his data interpretation was catastrophically flawed on every single level.
Yeah, he based this massive claim on a sample size of just 12 children. And even worse, there was absolutely no control group. He had no healthy kids to compare his subjects to. And later investigations proved he literally falsified the patient's medical histories to manufacture the timeline he wanted.
The paper was completely retracted and he lost his medical license, but the damage was done. People treated a tiny, uncontrolled, fabricated case series as if it were a rigorously controlled study like James Lynn's scurvy trial. It fueled measles outbreaks for decades.
Then you have cases where scientists actually discover something true, but their data handling is so unethical it corrupts the process.
Yeah.
Robert Millikan's famous oil drop experiment in the early 1900s is a prime example of this.
Oh, right.
He successfully measured the charge of an electron, which is a massive scientific achievement. But historians later read his private lab notebooks and found he routinely excluded data points that didn't perfectly align with his expected result.
He cherry-picked. He published his paper claiming he had included every single observation, when in reality he was tossing out the messy data that naturally occurs in any physical experiment. Even though his final conclusion was correct, discarding inconvenient outliers is scientific malpractice. Real data is inherently noisy.
It's like someone flipping a coin 1,000 times. If I come to you and say, I got exactly 500 heads and exactly 500 tails, it sounds completely correct in theory. But in the real world, probability is messy. You'd expect like 512 heads and 488 tails. Getting exactly a 50-50 split on 1,000 flips is highly suspicious.
This raises an important question, and that exact suspicion applies to Gregor Mendel, the monk who essentially founded modern genetics.
The pea plant guy.
Yes, the pea plant guy. In the 1860s, he bred thousands of pea plants and established the classic 3-to-1 ratio of dominant to recessive traits. His theories were brilliant and entirely correct. But decades later, the famous statistician R.A. Fisher analyzed Mendel's original data tables.
And Fisher found that Mendel's numbers fit the theoretical 3-to-1 ratio perfectly. Statistically, it was far too perfect. Given the natural variation of biology, it was mathematically implausible for his raw counts to land exactly on those theoretical targets without some kind of subconscious sorting or selective counting.
This is known in data interpretation as the too good to be true problem. When data matches a hypothesis without any of the messy variance we expect in nature, it is a massive red flag. Exam writers love to test this concept because it assesses your critical thinking. They want to see if you have R.A. Fisher's skepticism.
And that skepticism is crucial because of the reproducibility crisis. The sources mention a massive 2015 project that tried to replicate 100 published peer-reviewed psychology studies. More than half of those studies completely failed to reproduce the original results when another team ran the exact same experiment.
Which is pretty alarming. If you're going into clinical practice, you have to understand that a single data table from one study is never the final word. To protect yourself, you have to recognize the common misconceptions that lead to these errors. The most frequent trap on any science exam is assuming that correlation equals causation.
Oh, definitely. Just because two variables rise together on a scatter plot does not mean one caused the other. The classic textbook example is ice cream sales and drowning deaths. If you plot them on a graph, they both spike dramatically at the exact same time. They have a high positive correlation coefficient.
But eating an ice cream cone doesn't cause you to drown.
Exactly. The hidden third variable, the confounding variable, is the summer heat. Hot weather causes both a spike in swimming and a spike in ice cream sales.
Another trap you will absolutely encounter is axis manipulation. A student looks at a line graph on a test and says, wow, that line is incredibly steep. The change over time must be massive. But you can make a tiny 1% increase look like a vertical cliff just by zooming in on the axis and starting the scale at 99 instead of 0.
I always explain axis manipulation like looking into a funhouse mirror at a carnival. Your actual physical height hasn't changed at all, but because the glass of the mirror is warped and stretched, the reflection tells a totally different, exaggerated story. The data is the person, and the axis is the mirror. Always check the numbering on the mirror before you believe the reflection.
That's a great visual. You also have to separate precision from accuracy. People use those words interchangeably in daily life, but in data interpretation, they measure entirely different things. Precision is purely about consistency and repeatability. Accuracy is about closeness to the actual truth.
I like to think of it like throwing darts at a dartboard. If you throw 3 darts and they all hit the exact same spot in the top right corner of the wall, Your throw is highly precise. You did the exact same thing 3 times. But because you are nowhere near the bullseye, you are completely inaccurate.
Right. And in a hospital, you might have an uncalibrated digital thermometer that reads exactly 36.482 degrees Celsius every time you use it.
Mm.
That decimal implies incredible precision. But if the machine sensor is broken, the reading is totally inaccurate.
And speaking of broken sensors, that is the only time you are allowed to deal with outliers. An outlier is a data point sitting way outside the normal trend. The temptation for students and researchers alike is to just delete it to make the graph look cleaner, just like Millikan did.
But you should never delete an outlier unless you have a documented, defensible reason. Like knowing for a fact that the patient moved during the MRI or a specific sample was contaminated. Without a documented error, that outlier is part of the reality you have to interpret.
So we've walked through centuries of history, from Francis Bacon to the Challenger disaster. We know the mechanical traps. But let's bring this directly into your world. Beyond passing the TEAS or the HESI, how do you apply this? In nursing, you aren't just memorizing anatomy, you are constantly reading trend graphs. You are tracking vital signs, blood sugar levels, and medication responses.
Your ability to distinguish between a gradual linear change in a patient's heart rate over a 12-hour shift versus a sudden exponential spike dictates your clinical intervention. One might be a normal response to waking up, while the other might be a life-threatening cardiac event.
We also just lived through a masterclass in public data interpretation with COVID-19. Suddenly, everyone had to understand exponential case curves. But more importantly, the sources show how the media had to introduce logarithmic scales to the public.
That is a crucial application.
Yeah.
A standard linear y-axis goes up by set amounts, like 10, 20, 30. If a virus is doubling every day, a linear graph just looks like a terrifying vertical wall.
Yeah.
It's almost impossible to read the daily changes. A logarithmic scale changes the axis, so each tick mark is a multiple. 10, 100, 1,000, 10,000.
It essentially takes an exponential curve and visually flattens it into a straight line. By doing that, public health officials could easily see if the rate of infection was finally slowing down, rather than just staring at a vertical cliff.
You see the exact same critical thinking required when looking at climate change data. Bad actors will selectively choose a very short date range, say a specific cool period between 1998 and 2012, to make a long-term global warming trend look completely flat. They're exploiting the axis manipulation and cherry-picking tactics we just discussed to obscure the massive century-long exponential rise in temperatures.
So what does this all mean? Let's do a quick, practical sniff test. Say I'm scrolling social media, and I see a targeted ad for a new fitness supplement. It features a bar chart showing massive muscle growth, it proudly says, backed by clinical research, and it looks incredibly official. How do I dismantle that ad using the exam tools we just learned?
If we connect this to the bigger picture, it's the perfect test of your new superpower. First, check the sample size. Did they test this on 1,000 athletes or 12 guys in a basement? Second, look for the control group. Did half the group take a placebo pill while keeping everything else identical, or were there confounding variables?
Right, like maybe the group taking the supplement was also forced to lift heavy weights for 3 hours a day, which is the hidden third variable actually causing the muscle growth.
Exactly. Third, check for the funhouse mirror. Look at the axis on their flashy bar chart. Did they start the axis at 0, or did they start it at 98% to make statistically insignificant 1% gain look like a towering skyscraper of muscle? Finally, are they claiming strict causation, saying this powder unequivocally caused the growth, when their messy data only really shows a weak correlation?
The same exact skills you need to ace a nursing entrance exam are the exact same skills you need to not get ripped off on the internet. Let's summarize the ultimate takeaways for your test day. Always identify your x-axis as the independent manipulated variable and your axis as your dependent measured result.
Remember that a cloud of points is a valid finding of no relationship. Correlation never guarantees causation.
Always check the numbering on your axis before judging a trend's steepness. Understand that precision is hitting the same spot, while accuracy is hitting the bullseye. And never discard an outlier without a documented reason.
If you internalize those rules, you aren't just memorizing definitions for a multiple-choice test. You are actively shielding yourself against misinformation. You are transitioning from that 17th century scholastic mindset of just letting someone else tell you what's true into a modern empowered mindset where you evaluate the raw evidence yourself.
It really is a superpower. You learn to cut through the noise, spot the manipulation, and find the actual truth hidden in the static of the real world.
And that leads to a final thought I want to leave you to mull over. Think back to John Snow, mapping those cholera deaths in London. His ability to understand reality and ultimately stop a deadly disease was entirely dependent on what he chose to measure and plot on a physical map. Our understanding of the universe is fundamentally limited by what we can measure. So if reality is bound by what we graph, what vital, invisible variables in our daily lives are we completely misinterpreting right now simply because we haven't figured out how to put them on an X and Y axis yet?